Okay, guys, I'm back. I hope you guys had a good Thanksgiving. I've already gone over chapter 20. It's up on YouTube. I'm going over chapter 21 today. And then this shouldn't take that long because there's not a whole lot in here. But I will go over chapter 22 with you guys on Monday and answer any questions. We'll take Monday to do all that. And then Tuesday we'll start up with chapter 23. All right, 22 is going to be short and it's going to be a lot easier to get because it's just Introduction to Evolution. So I'm not going to go over it because it's a pain in the butt to try and put these on YouTube. So let's get started. Okay, so in chapter 20 we talked about different types of technology that you can use to study genomics and a little bit of proteomics. Now we're going to go over how you apply those technological processes to try and figure out the development of an organism. So in all organisms, all right, especially in the mammals, okay, but I mean some invertebrates too. You always start with a single-celled zygote, okay, which is a fertilized egg, right? After sperm and egg meet, they fuse, the nuclei fuse, and now you've got one zygote, okay? So now, the zygote is going to go through 3 steps, all right? Cell division, cell differentiation, and morphogenesis. All right, cell division just means that after the zygote's formed, it's going to go through a series of rapidly dividing, basically mitosis, and it's going to increase its cell count, all right, significantly and very fast. Secondly, after it started to divide a lot, it's going to have the precursor cells necessary for all of organ functions and differentiation and stuff like that. So it's going to start differentiating, which really means that it's going to have specific structure and function to fit its tissue needs. Thirdly, it's going to start orienting its organism into a specific shape. So you have a posterior end, anterior end, dorsal ventral. You know where this organ is situated, this one is situated, and that's called morphogenesis. Okay, basically it means body formation. They overlap. All right, so the cell differentiation and morphogenesis especially are going to overlap. So your cells are going to be differentiating while they are being positioned in the developing organism. Now, we don't use a lot of organisms to study development. We use the ones that are easy. Now, we've already kind of talked about what the criteria are for model organisms. They have to have a short life cycle. They produce a lot of progeny. You can see readily observable phenotypes if you're studying for traits in Mendel's case. But for development, in terms of studying development, we need to be able to see the embryo. Okay, so first of all, you have to have a really observable embryo. Okay, case in point would be the ones that birth their embryos out, and then their embryos develop in the environment, in the external environment. They have to have a short generation span. They have to have relatively small genomes. Okay, large population of progeny, and it helps to know something about the organisms before we study them. Okay, so specifically I'm describing the ones that are like C. elegans, okay, with the nematode. Drosophila, which is the fruit fly. We've also used zebrafish, they use mouse, and luckily we've already sequenced all those genomes, so we know a lot about them already, genetically. Now we're going to look at them developmentally. So, there's a lot of pros and cons for using specific model organisms. They're on page 405. Okay, and it goes through some pros and cons of using, Drosophila versus the C. elegans and all that. I'm not really going to go over the pros and cons. You're just going to know that these are the ones that we use a lot of, and I'm going to go over specifically which organism you would use to see what. Okay, so we would use Drosophila to see morphogenesis and to see how the organs are spatially arranged, but we wouldn't use we would use the C. elegans to see how the vulva develops and how cells talk to each other to position themselves. Okay, so model organisms are good to use not just for every single thing, but they are good to use for specific things. All right, so that's what we're going to talk about today. All right, now, the great thing about C. elegans, and I'm not just saying this because I was a C. elegans researcher, but because I this is what the scientists say is true. C. elegans is going to be good to use if you're looking at cell lineage. All right, so basically what cell lineage means is that all cells start from one cell, all right, from the zygote, and then they start to rapidly divide. Well, C. elegans lays its eggs into the environment, and they're easily seen underneath a microscope, and we can tag them to see which one comes first and how they divide and how they spatially are arranged. So what we'll do then is we'll trace the cell lineage, okay, which basically means it's self-explanatory. You're going to see where each cell came from previously. All right, so if I have one cell, it divides into two, I can pick which one and then trace that lineage. So it's going to divide twice and then divide twice and then divide twice, and we'll be able to see where every single cell originated from, okay? Now, this becomes important when we're trying to look at which cells become what, all right, the fate of those cells. And we would map it out and call it a fate map, all right? Go figure. Now, most of the time, cell differentiation doesn't mean that the cells look any different, okay? All cells have all of the genetic material that they would need to become any kind of cell, which is what a stem cell is. It's which genes are going to be turned on and turned off that is going to tell you which one is going to become the kidney, or the heart, or the lungs, or the abdomen, the thorax, the wings, the whatever. So really, it's all about transcriptional and translational regulation, right? Controlling gene expression, basically. Now, how do they become inactive then? In the case of animals, all of our cell differentiation and morphogenesis happens by the time we're born, in the embryo, basically. In plants, though, there are some parts of the plant that actually will go through, they have stem cells throughout their entire lifetime. Case in point that they're going to talk about in the book is the apical meristem. All right, and in this case, these cells are at the tips of the roots, and they have the ability to become anything, to replace any kind of organ in the plant. Now, we have some latent, not totipotent, but probably like pluripotent or multipotent types of stem cells, the ones that can become most things but will not become everything, doesn't have the ability to become everything. And that is rare for us to see. That, I mean, we don't make nerve cells, we don't make brain cells. We can replace different differentiated organs already, like your liver can regrow and things like that, but it's not as widespread as in